Hey everyone, this is Miss Segovia and I'm going to walk you through the semester exam review sheet. I'm going to go pretty fast because there's a lot to cover and I've got limited memory on my cell phone. Big thing to know though is that we also have a PowerPoint at tinyurl.com/fall exam review of 2015 and you can download that PowerPoint to view it and get a lot of great information there. So let's go through here. To begin with our biomolecules, carbohydrates, lipids, proteins, and nucleic acids are polymers, large molecules made up of smaller subunits known as monomers. Complete the following table, and you can use page 166 to 171 to help. But here we go. Carbohydrates. CHO are the elements, and you usually see them in a geometric pattern. The monomer is glucose, or really anything that ends in the word "ose." We use them for quick energy and breads, pasta, and sugars. Lipids are also CHO, but when you see these, you'll usually see a line of carbons with a whole bunch of hydrogens. This is because lipids have lots of long-term energy. Remember, anytime you see lots of bonds, that means we're talking about lots of energy. There might be an oxygen on there somewhere too, so just kind of keep that in mind that you will see CHO, but tons of carbons and hydrogens. Kind of look like little train tracks or like a centipede. Fatty acids and glycerol are the monomers, and again, like I said, the function is long-term energy storage. So I'm not fat, I've just got lots of energy for later. Examples are butters, oils, and fats. Proteins are CHON S, oxygen, nitrogen, and sulfur in there. And the monomers for this is an amino acid. The building blocks of all organisms, we use them for enzymes, meat, bones, and hair. When you see these, you'll typically see the pattern NCC. That's kind of the dead giveaway that you're looking at a protein right there. Nucleic acids are CHON P instead of CHON S, and these we'll see, it's really cheesy, but I kind of picture it like a chubby little kid holding a pistol. So he's going to have the little base right here. It's like pew, 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 pew. And then on the other side, he's got a little phosphorus balloon. And so we've got this little sugar unit in the center with a phosphorus and a base. Wait, we love DNA made of nucleotides, sugar, phosphate, and a base bonded on each side. Yep, I just did that. So they carry the genetic code, and the examples are DNA, RNA, mRNA, tRNA, all the good stuff. So that's your quick on the biomolecules. Prokaryote versus eukaryote. In prokaryotes, there is no nucleus. That's your huge thing to know on prokaryotes. No nucleus. Eukaryotes, they have a nucleus, they have organelles, they tend to be big, these guys tend to be small, so on and so forth. Between them, they both have ribosomes, they both have DNA. This one just has no nucleus, this one does, and they both have plasma membranes. That's kind of your big thing right there. Again, pro means no. You do have a nucleus. So, key elements to remember on that one. With levels of organization right here, we go, they give us our options, organ system, organelle, tissue, population. So here we go. Atom, molecule, blank, and cell. When in order to have a cell, we gotta have organelles. Then cell, blank, organ. And so organs are made out of tissues. And then organ, blank, organism. That would be organ system. And after organism, our only one left is a population. And then we have our wonderful list of organelles. So a nucleus. If we have a great big cell and you see something inside it, that is the nucleus, and inside there is going to be a whole bunch of DNA. So let's do the pictures real quick first. Our cell and plasma membrane, if we have our great big cell right here, we see the outside is our plasma membrane, and if we zoom into that, you're going to see these little phospholipids with all their little tails pointing to the middle, something like that. A cell wall, so if we got our cell outside the cell, especially if you're looking at a plant cell, you've got a thicker area there. That is the cell wall. That provides shape, support, and protects the cell. Ribosomes, they look like these funny little things. You'll see them like this if they're drawn individually, or if we're looking at a cell, they're the little dots that are inside the cell. Cytoprotein synthesis, mRNA uses A and U, and then C and G. The Golgi body, when we see the Golgi body in the cell, it tends to look kind of like a lava lamp, and you get these like weird little things, and then you get these little droplets coming off and kind of crazy stuff like that. So that Golgi body with all the little droplets coming off, it sorts and packages proteins, because remember, it's all about making those proteins. Mitochondria tends to look like a weird little jelly bean that's got like some squiggly line in it. It converts glucose and oxygen into carbon dioxide, water, and ATP, which is energy. Because we need that energy to keep us going. Like, you got to keep going through this packet. Chloroplast. When we see a chloroplast, it kind of looks similar to the mitochondria. If you're lucky and you get one in color, it'll be green. But we tend to see an organelle that's got like these weird little stacked pancakes inside it. So it contains chlorophyll that absorbs solar energy to be used for photosynthesis. Got to write tiny. And makes oxygen and C6H12O6, better known as sugar, and sometimes known as organic molecules. So now if we go back up here, let's see where everything is. So we got to put a check mark if we find it in those things. So nucleus, we do not find in prokaryotic cells, but we do find it in animal, and we do find it in plant. A cell or plasma membrane, we find in prokaryotes and animals and plants. Cell wall, we will find those sometimes in our prokaryotes, and we also find them in plants, but we do not find them in animal cells, because otherwise you'd be stiff as a board. You have to move around. Golgi bodies, that's an organelle, so we don't find it in prokaryotes, but it is in animals and it is in plants. Our mitochondria, again, another organelle, not in prokaryotes, but it is in animals and it is in plants. And finally, chloroplast, also an organelle, so it's not in prokaryotes, but it's not in animals either, but we do find it in plants. I wish we could do photosynthesis. Get some sunshine, get some energy, go home for the day. You go on a diet, you just go in the closet, be awesome. All right, cellular transport. Passive transport does not require energy or ATP. Examples are diffusion, facilitated diffusion, and osmosis. Active transport requires energy, ATP. Examples are the sodium-potassium pump, endocytosis, and endocytosis again. I think we meant for that to be exocytosis. So here we'll put a little exocytosis, bringing things in and letting things out. So during osmosis, water enters or leaves the cell through the cell membrane. The water moves from a high concentration to a low concentration. It literally flows downhill. Your body is constantly regulating your internal environment, or homeostasis. On a hot day, how would your body maintain homeostasis? Can't talk because I'm so exhausted. I'm sweating. No, really, I'm not. So sweating allows for evaporative cooling. As that sweat evaporates off your body, it drops your temperature. It's awesome. Explain why the boy almost died when he drank all that soy sauce. Well, what was happening was too much salt, and it was causing his cells to shrink, causing his blood cells. And when those guys start to shrink, you can't carry oxygen. So no O2 equals death. You like my notes right there. So too much salt, cells are shrinking. In fact, it would have looked a little bit like this if he were a plant. And so label each of these cells as hypertonic, hypotonic, and isotonic. So in this first one, we can see there's equal water in, equal water out. That is, and I'm just going to put the first three letters, isotonic. Here we got tons of water going in. This thing's getting fat like a hippo. So that is hypotonic. And this one's shrinking down super skinny because it's super hypertonic solutions right there. All right, cellular energy, photosynthesis and cellular respiration. Photosynthesis is a transfer of energy from solar radiant energy to chemical energy because we're turning it into glucose. Plants and algae can do photosynthesis because they have blank, which is able to capture the solar energy from the sun and then convert it to chemical energy. That is chloroplasts. Cellular respiration is a transfer of energy from blank energy. Oh wait, glucose, that was chemical energy, to a different form of energy or ATP, and heat also released. Remember those posters that we have up around the room? They're great inspiration. So the equation for photosynthesis is that we have to do with their plants. We got to breathe on them. We got to give them CO2. We got to give them H2O, and then we got to give them some sunlight. And what do we get out? We get out C6H12O6, also known as sugar or glucose, and then they also are kind enough to give us oxygen so we can breathe and not suffocate. Thank you so much, plants. Cellular respiration. You guys go down to lunch, and instead of buying healthy food, you buy those slushies and you get tons of sugar inside of you. And you also have to breathe and bring that oxygen. You give off CO2 and H2O, and we also get a little bit of ATP energy that we can use and burn, which is why you're so hyper when you come to class after lunch. So mitosis versus meiosis. Remember, the cell cycle represents the entire life of a cell. The cell spends the majority of its time in interphase and a short period of time during mitosis. After mitosis, 2 new daughter cells are formed. Each daughter cell has the same number of chromosomes as the original cell. So in mitosis, you get the same number. You gotta know that one. If you start with 6, you end with 6. If you start with 23, you end with 23. And so here's an awesome picture. We started with 2N, we ended with 2N. Awesome. How many daughter cells are at the end of mitosis? 2. If a parent has 6 chromosomes at the beginning of mitosis, how many will each daughter cell have at the end of mitosis? Again, it's the same. So 6. Did the number change? No, it's the same. Explain what happens if a cell becomes cancerous. It divides out of control. It just goes crazy like I am trying to finish this while I still have memory in my phone. DNA replicates during the S phase of the cell cycle. Fill in the complementary nitrogen bases to this segment of DNA. So we've got all these, and so our A is going to bond, the DNA. So if we're going from DNA to DNA or DNA to mRNA. So A is going to bond with T, A is going to bond with T, C with G, C with G, T with A, G with C, C with G, T goes to A, and A goes to T. Over here, though, if we're going to turn it into mRNA, A is going to bond to U instead. So AU, AU, CG, CG, TA. Then we got G to C, C to G, T to A, and A to U. So that's your big clue right there. If it's a T, it's DNA. If it's a U, it's mRNA. Refer to a codon chart and translate the following mRNA into amino acids. So the first thing you're going to want to do is divide it into sets of 3 because we always read in a triplet codon. Then you're going to want to go to your handy dandy notebook, which I'm doing right now, where you can find your codon chart. So just a reminder on how to read these codon charts. Our first one is AUG. So let's look. Our first letter is A. Our second letter is U. Sorry, there you go. AU, so that means we're in this box. And so our last letter is G. We see right here, third base. That is methionine. So right here, I'm going to put MET. Next one is UCC. So let's see, we got U, we got C, and then another C. That's serine. So I'm going to put SER for serine. AGC, letter A, G, C. Oh, it's serine again. But notice we're using a completely different code and still getting serine. So you always got to check your codes. AGA, so AGA was really close. Look at that. AGU was serine, AGA was arginine. So I'm going to put ARG. One little mutation could throw the whole thing off. And then UAA, so UAA, stop. Yes, thank you. I absolutely will. All right, so we put a stop in right here. So met, sir, arg, stop. All right, after meiosis, the new cells will have half, look at Meiosis was half. Remember, mitosis was same. Half the number of chromosomes as the original. So we start out at 2N, we end at just 1N. According to the diagram above, how many cells are at the end of meiosis? There are 4. If a gamete, egg, or sperm has 6 chromosomes at the beginning, how many will it have at the end? It has half. So it's only going to have 3. Did the number change? Yes. Mutations that can be inherited by the offspring will occur only in the gamete cells. That's your sperm and eggs. If you have a mutation in your arm, your kids don't care. Mutation in your eggs, ooh, that can cause trouble. Why does crossing over occur? Remember, that's when they're all lined up and then they'll switch and they'll like pass off other parts of DNA to the neighbor. And so crossing over occurs so it allows for more variation or variety, gives us more options in our offspring and what's make you super unique. All right, protein synthesis, fill in the missing steps. mRNA transcribes the blank within the nucleus. It transcribes the DNA within the nucleus, and instead of using the base T, we're going to use the base U, so we put uracil. The blank leaves the nucleus. We lose our mRNA. It can exit the nucleus to join up with a ribosome. The ribosome will blank the 3 bases to identify the blank blank. All right, so it's going to translate the 3 bases to identify the amino acids. That's what that whole chart was about. Once the chain of amino acids is completed, many will then go to the blank to be packaged and shipped. Our own little UPS, which is the Golgi. Golgi body, Golgi apparatus, Golgi system, whatever it is, it's still the Golgi. Genetics! Oh my gosh, I think this is it. This is our last page, isn't it? Please? Yes. Okay. Purple flowering plants alleles are dominant to white flowering alleles. Create a heterozygous purple flowering plant with a homozygous dominant flowering plant. So we're going to cross those. Sorry. So heterozygous means we're going to have one big, one small. So it's a big P and a little p. And homozygous means they're going to be the same. Dominant means it's capital. So big P, big P. So let's go ahead and write these out. We got big and little for one parent, and then we got big, big for the other. So our offspring on our Punnett square could potentially look like this. What's the chance of purple plants? Well, let's see. If they have a dominant, that means they're going to be purple. And oh my gosh, all of them have a dominant. So that means we have a 100% chance of them being purple. So which means none of these have the possibility of being white. The chance of them being heterozygous means they're going to have one big P and one little p, and we have 2 out of the 4, so that is a 50% chance. Dihybrid. In pigs, curly tail is dominant over straight tails. Pink is dominant over gray. Below is a cross of a male pig that is homozygous dominant for both traits with a female pig that is homozygous recessive for a type of tail and heterozygous for body color. And so if you can't understand that, the nice thing is they gave it to you here. So homozygous dominant, homozygous recessive, and heterozygous. And the table's been all filled out for us. So out of our 16 possibilities, what are the chances of having a pink pig with a curly tail? So again, in order to be pink, we need to see a capital P. In order to be curly, we need to see a capital C. So let's count how many we see with that. We got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. All of these have a capital C and a capital P, which means we have a 16 out of 16 chance. What are the chances of having a gray pig with a straight tail? So in order to be gray, they'd have to have 2 little p's. In order to be straight, they'd have to have 2 little c's. And so if we look through here, we don't have any that have just little p. And if we look for our pink color, that means that, or I'm sorry, gray with straight, so straight tail, little c. Do we have any here that are going to have a straight tail? None. There's a 0% chance. None of them have the recessive traits for both alleles. So each one has a capital. Now they want us to try our own Punnett square, where we're going to cross a heterozygous with another heterozygous. Oh, this is so much fun, right? No, I hate these. All right, so we've got big R, big Y, big R, little y, little r, big Y, little r, little y for one parent. And then we've got the other one, little, little big, and little big. So remember, if the parents start out with 4, all the offspring have to have 4. And so this little diagram here is showing you they can give 1 R and 1 Y, and so you just have to find all the different combinations. So I'm going to hush up real quick and do these real fast. And you could follow along, or pause the video, work it, and then go back and look. I don't know why I put a P right there. Let's pretend for a moment that this is for round and yellow. And so how many of ours would be round and yellow? That means we'd have to have a capital R and a capital Y. And so we'd have 1, 2, 3, 4, and then 5, 6, 7, 8, 9. We would have 9 out of 16. And then how many would be round, and let's just say that other one would be green. And so that would have to have a capital R and a little y, 2 little y's actually. And we can see we've got 1, 2, 3 like that. So that's 3/16. And how many would be, let's assume our other trait is wrinkled yellow. So that means little r, big Y with something else. So 1, 2, 3. And then how many would be wrinkled green? That would be little r, little y, little y. Sounds like an auctioneer. And we only have 1/16. And so, of course, there's a super easy way to do that. You can make it much easier on yourself. And that's just to do 2 small punnets. And so for the big R, little r, big R, little r. And then do a second punnet that uses just the Y's. And same thing, if I wanted to know how many were round and yellow, so if I wanted to know round and yellow, let's see, we've got 1, 2, 3 fourths, and yellow 1, 2, 3 fourths, and I would multiply those two together, and I would get 9/16. Oh, that's so much easier, takes so much less time. Let's see, how many would be round and green? We got 1, 2, 3 are round, so that's 3/4. Only 1 would be green, this little guy right here. So that's 1/4, and so that is 3/16. And you end up with the same answers if you do the mini punnets instead of the giant punnet. So trust me, do the mini punnets. All right, folks, that concludes your semester exam review. If you have any questions, please ask me ASAP. I'm happy to answer, and I hope you found this somewhat helpful. Thank you.